I'm extremely happy to have Jennifer Barrow today for our Limno seminar series for this October month. Jennifer just finished her master's from McGill University. Before that, she did a bachelor at Queen's University in John Small's lab, and she published a research paper investigating diatom dynamics in South Central Ontario, trying to answer this question about calcium decline and acidification. And you can find Jennifer's paper in the Journal of Paleolimnology, published in 2014. Jennifer also did a Bachelor of Education at Queen's, and she did teach for a while before returning to graduate school. She did her master's with Rini Gregorieves at McGill, co-supervised by B. Beisner at UQAM in the context of the NSERC Create ECOLAC. So NSERC is our federal funding agency that also has this very extensive training program, and Jennifer was part of one of those for her master's. And I think I made a mistake. Jennifer, I'm sorry, on the title of your presentation. Which actually will be, I think I have it here. Community responses to macrophyte abundance and nutrient loading in experimental ponds. Is that correct?
That is correct.
And Jennifer, please start sharing your screen, and the floor is yours.
Thank you. Let me just get this set up. Okay, how does that look?
So far, I see you.
Oh, you do not. Sure. Okay.
Very good.
How about now?
Perfect.
Okay. Okay, thank you for the introduction, Alex, and thank you everyone for joining in on this SEL Limno seminar. I also want to say a quick shout out to my parents and my siblings and my friends who are also out there listening. Hey, and thank you for the support. So as Alex said, my name is Jennifer and I'm a master's student at McGill University. And today I'll be presenting research on the response of two groups of microorganisms, phytoplankton and bacteria, in experimental ponds. So there's a huge interest in microorganisms because they play essential roles in many processes. They are everywhere. In humans, it's been found that the body is made up of more bacterial cells than human cells. We rely on microbes to function efficiently. And there's also research into how we can use signatures of microorganisms to be indicators of life on exoplanets, as they can live in extreme environments and indicate the presence of a productive space. And this is what the figure on the right is pointing to. So studying microorganisms can help us understand the system, manage a system, assess it, and see if it's working. So then it's only obvious that the importance of studying microorganisms in fresh water, as water is a thing we need to survive and it's what makes Earth habitable. So my focus is on shallow lakes, and there are many definitions for a shallow lake, and I work with the definition that it's a body of water where light can penetrate to the bottom, which allows macrophyte growth. Shallow lakes are the most abundant lake type globally, and they support a diverse fauna and flora and provide many ecosystem services, such as recreation, irrigation, drinking water, and contribute to our global carbon cycle. They are important ecosystems, and yet these systems are highly sensitive to disturbances. Things like cultural eutrophication, which is often the result of urbanization and development, is a major threat to water quality and biodiversity. As you can see in this figure, high nutrient loading can cause a clear shallow lake to be something dominated by algae, green from top to bottom. And these algal blooms can also be toxic, another reason why we don't want eutrophication in our systems. These are two alternative shallow lake states, clear and turbid, and there's a lot of research into this phenomenon, but there are still gaps into our knowledge of why a lake and when a lake will shift to be turbid. So this phenomenon leads into a critical transition, which occurs when the lake shifts from a clear state to a turbid algal dominant state, or vice versa. In the figure on the right, on the x-axis is nutrient concentration, on the y-axis is turbidity, and the dashed line is a threshold. Below the line, the lake state is clear or blue, and above it shifts to a turbid state. We can see that as we increase the nutrient load, there's an increase in lake turbidity. This figure also shows that lakes can recover from eutrophic conditions if the nutrient load is reduced. However, the recovery takes a different path. It's very unlikely that the lake will go back to be the exact same before eutrophication. And the reduction of nutrient is a costly, expensive, and time-consuming process. Another concept of the alternative stable state model, this clear and turbid conditions, is that the behavior of a lake is nonlinear. The area in the middle, as shown in the figure on the right, is hysteresis, as when given the same conditions, such as the amount of nutrients, either stable state is possible for a said lake. So there may be many factors that play into whether or not a lake will become one state or the next, and one of these is the presence of macrophytes. As you can see in this figure, macrophytes are the aquatic plants at the bottom. So our macrophytes, our aquatic vegetation, can mitigate the effects of nutrient additions. In shallow lakes, macrophytes are a part of a positive feedback loop which promotes clear water conditions, and they inhibit phytoplankton growth via various mechanisms such as using nutrients, shading, and increasing sedimentation. If we look at this figure, we can follow along one of these processes. So vegetation there in the center has a negative impact on nutrients, and nutrients has a positive impact on algae. To get the impact of vegetation on algae, we multiply those two signs. So a positive times a negative value equals a negative value. So vegetation has a negative effect on algae. So vegetation inhibits algae growth. Following different loops, it's apparent that vegetation positively supports vegetation, and algae and turbidity positively supports turbidity. So these two states, clear, vegetated, or turbid and algae, are stable on their own. So I want to investigate more this interaction between vegetation, nutrients, and algae, as well as incorporate some ideas around, or some information around bacteria, so that we can better understand the influence of macrophytes on microorganism communities. And it's important because of the cascading effects it can have up the food web, as well as it can dictate how the ecosystem functions and what stresses it can endure. So this brings me to my research. The goals of my research projects are to characterize the relationship between phytoplankton, macrophytes, and nutrients in shallow lake systems, and to characterize the bacterial communities in respect to macrophyte abundance in these same shallow lake systems. So to do this, looking at just the phytoplankton, I wanted to measure response in two approaches. I looked at a traditional taxonomic approach. And so in my water samples, I identified and enumerated what I saw. And I also took a functional approach, which takes into account phytoplankton size, shape, and their abilities. The functional approach can be more ecologically relevant, and studying both increases our understanding of the system. For the bacterial communities, I looked at two habitats within a pond. I looked at the free living community, which interacts with phytoplankton in the water column. And I looked at the attached community, or epibiotic, which was associated with the macrophytes in our pond. So that means they were interacting with the macrophytes as well as phytoplankton that would be on the macrophyte leaves. It's important to note that the water does not act as an isolating barrier to these two communities. Epibiotic bacteria can diffuse into the water column, and the free-living bacteria can be a source pool for the epibiotic bacteria. So though I'm looking at two different communities, it's likely that we can see overlap when I'm looking at diversity measures. So it'll be interesting to look at how distinct they are. Right? One way to study freshwater ecosystems could be to go out to lakes and sample a bunch of shallow ponds. However, this can make it hard to disentangle what factors are interacting with the system to cause the differences we see. Instead, we can turn to using experimental system. We use experimental ponds, which would allow us to design a system that we wanted and manipulate and monitor it with relative ease. Research has shown that accurate models of the physical and biological dynamics in shallow lakes can be observed accurately in experimental systems and applied to freshwater lakes, shallow lakes. Using experimental models to study phytoplankton growth and the response to ecosystem stressors has become a popular scientific approach during the last 3 decades. However, many of these studies looking at the effect of nutrient additions only measure phytoplankton response using chlorophyll A concentration. And this does give a detailed insight onto how the community, onto how the phytoplankton in total has responded to a certain stressor, but it doesn't give us any information into how the community is changing and which impacts ecosystem functioning and also a resilience of that system to a disturbance. Of the studies that examined community composition, most did it for one time point and very few investigated functional composition, which I'm looking to investigate or which I investigated. So there's a gap in the understanding and available literature on dynamics of microorganisms at the taxonomic and functional community composition. And so there's a need for more work, which is why I'm here. And so I will briefly explain our pond setup and then go right into our results. So our ponds were in large plastic tubs, which contained 350 liters of water. We added sediment, phytoplankton. Sorry, now you can see. So this is kind of like what our pond looked like. They contain 350 milliliters of water. We added sediment, phytoplankton, zooplankton, and a juvenile rock bass, which is a planktivorous fish. After we added these into our ponds, we then gave them a treatment. Our treatments were either 1 of 3 macrophyte treatments using Alodia canadensis as their species. Ponds either had 0, none, which was 0 or no macrophytes, none. Oh, I'm saying the same thing. Sorry. Ponds either had 0 macrophytes, a low amount, which is 30, or a high amount, which was 120 stems. And the second treatment was the nutrient treatment. Ponds received nitrogen or phosphorus once a week or once every 2 weeks. And the nutrient load equated to 10 micrograms per liter of phosphorus and 200 micrograms per liter of nitrogen. During our pond experiment, after we had the setup, we collected data. We collected data daily and some data weekly. Daily, we collected samples for chlorophyll A analysis, so we can get an idea of the concentration of chlorophyll. We measured dissolved oxygen, temperature, conductivity, and pH. And weekly, we collected water samples for total phosphorus and total nitrogen analysis, for phytoplankton analysis, and zooplankton analysis. And you can probably tell that this project took a whole team to run. So this is a picture of our mesocosms, of our ponds, our mesocosms. The experiment was conducted outside on the roof of the biology building here at McGill. And we ran the experiment for 12 weeks or 76 days during the summer. So there are over 200 phytoplankton samples available for me to look at. However, that's quite a lot for one person to do for their master's. So for my phytoplankton analysis, I focused on samples from ponds that only received weekly nutrient addition. So this is a high nutrient level. And from this, I then would take a subset. And it's also important to note that I collected one bacteria sample, or a bacteria sample at one time point, which was day 76. So this is one of our first figures. It's a figure of the total chlorophyll concentration over the 76-day experiment in the ponds that received weekly nutrient additions. So we have time on the x-axis and total chlorophyll on the y-axis. This is using the daily chlorophyll A data. The no macrophyte treatments will always be in blue. The low macrophyte treatment will be in yellow, and the high macrophyte treatment will always be in green for my figures. And so for my phytoplankton analysis, as I mentioned, there's a lot of samples, and we made some educated guesses based on this figure to focus my time and energy onto a subset of samples that I would look at. Keeping in mind that we only have water samples at weekly intervals, I looked at samples from weeks 1, 4, 5, 7, 10, and 12, which are the experiment days indicated by the arrows, day 1, 20, 27, 41, 62, and the final day, 76. And on that same final day, I have my bacteria sample. Okay, so right into the data. I followed standard microscopy work, and even though I was coming from a background in diatom work, there was still a good learning curve for me to do phytoplankton analysis. But I successfully identified a lot of genera. My samples were diverse. There was 62 genera in the 54 samples. So I had the 6 time points, and at each time point, there's 3 replicates per treatment. So that makes the 64 samples. And after I collected my data on what taxa was in a community, and I can convert that into biovolume. And so that's the first step. And this is my figure here on the right, which is the total biovolume of phytoplankton among the 3 macrophyte treatments over time. So I have time on the x-axis. And it's very nice to see that my plot on the right using the count data is similar to what we measured using the chlorophyll A data on the left. We see that the high macrophyte treatment had a relatively stable phytoplankton biovolume throughout the entire experiment. We know that the addition of nutrients causes an increase in phytoplankton biovolume, which was expected, but we see that this growth, the phytoplankton growth, is modulated by macrophyte abundance. Circled here are significant differences in biovolume concentration. So for example, at day 20, we see that the biovolume in the high macrophyte treatment is significantly different than the biovolume in the no macrophyte treatment. And we see this again at day 27, day 62, and day 76. By doing the phytoplankton microscopy, I can determine if these differences in biovolume are results of an overall increase in everything, or more importantly, did some taxa become dominant in these nutrient-rich conditions, or did some taxa become dominant in one macrophyte treatment but not in another? One way that I can show this is by using a principal response curve analysis to plot how the communities changed over time as a result of a treatment. The y-axis is a measure of treatment effect. With the greater the absolute number, the greater the effect. And on the x-axis is time. And we can test the significance of this model too. And the one I will show you is significant. So I'm going to plot how nutrient loading affected the phytoplankton communities in the 3 macrophyte ponds. For a principal response curve, there needs to be a reference treatment, and we selected the high macrophyte treatment to be the reference, as it was the stablest at the chlorophyll level and showed the lowest responses. This reference is the straight line here on the figure, the green line, and the communities in the other 2 macrophyte treatments will be compared to the reference at each time point. And so how the community responded will plot either above or below this line. And here are the results. So this is how the phytoplankton communities in the no macrophyte treatments, which is the blue line, and the low macrophyte treatments responded over time. We see that the low and no macrophyte ponds followed a similar pattern. They both go up and slightly down in the community dynamics. However, in general, the response in the no macrophyte ponds was stronger than the low macrophyte ponds, and we see this because the curve is higher. And this is always in comparison to the reference. So the reference pond community can still be changing, is still changing, and it's how did the other 2 treatments ponds change relative to the reference. The principal response curve can also tell us the individual response of each taxa. And here are some of the major taxa identified. So we have chlorella, which is a small green chlorophyte, lobomonas, small chrysophytes, Nitzschia, diatom, a starastrum, Stardecimus complex, and Cryptomonas were all identified as responding to the treatment more strongly than the other taxa. The higher the weight, so this is where the taxa appears on the axes, looking at the treatment effect on the y-axis, the more likelihood the response curve that we see in the middle is likely to follow the pattern. Sorry, the response pattern of the taxa is likely to follow the pattern of the treatment curve. So I will walk through an example. If we take chlorella, we can loosely interpret these changes that at week 4, the change in chlorella biovolume in the no macrophyte treatment is sort of equal to the blue curve times the treatment effect of 0.6. So the point on the curve is positive because it is above the green line. And we multiply this positive value by the positive treatment effect, which gives a positive taxa response. So chlorella increased in biovolume relative to the changes that occurred in the reference pond. And so it could be a lot to interpret, but one of the things that you can take away if you just stand back and look is you see that 2 taxa for chlorella and cryptomonas have opposing treatment effects. And you can be wondering, why is it so? And so if we plot their change in biovolume over time, we may be able to see a clearer difference. And that's what I've done. So on the x-axis is experiment time, and on the y-axis is biovolume. So we have the figure on the left, which is for chlorella, and the figure on the right, which is for cryptomonas. And we see that these 2 taxa peak at different times, which could be why they have these different responses from a principal response curve analysis. We can also look at other taxa identified by the principal response curve. So we had the Starodesmus starosira response, and then Nitzschia was also indicated. So for Starodesmus starosira, they did not have a high abundance, but the response appears to be related to macrophyte presence, as they don't really increase in the no macrophyte treatment. And Nitzschia increased in all treatments over time, which could be why it had a response on the principal curve analyses closer to 0, because there was no treatment effect, it was more of a time effect. So now we can start to ask questions, why did some taxa increase at one time point and decrease in another? And this can go into a variety of reasons. When we compare the small green chlorella to the long Nitzschia, we may be implying a difference in role based on their size and shape. And this thought process leads nicely into the work on functional traits. So a functional trait can be based on morphological, ecological, and physiological attributes. Our functional approach adapted this table on the right. In this figure, there are different colors for 4 different types of traits: morphological, physiological, behavioral, and life history. And in the columns, there can be sorted into different ecological functions. There's reproduction, resource acquisition, and predator avoidance. Studying phytoplankton at this level can speak to the role of the taxa, and studying communities at this level can speak to the community's functionings and abilities. These are the trait matrices I looked at. So phytoplankton growth is limited by the rate at which they can acquire essential resources while evading predation by zooplankton. So for the 64 taxa in my phytoplankton dataset, I gave each of them a value, could be numerical, binary, or categorical, for each of these traits. Some of the traits, like pigment composition, silica demand, and capacity for toxin production, are rooted in phylogeny, but others are not. We can see that some traits appear in both matrices. For example, the volume of a cell, so AU volume, speaks to how much nutrients it requires, whereas it can also speak to if that taxa is edible or not, and that would give it to the herbivore interaction. A taxa that produces large colonies or has a maximum length greater than 35 micrometers is likely to be inedible to zooplankton. So before I present my work on traits, I'm going to just show you, go back one step to mention that one of my focuses was to look at diversity and how diversity changed in my macrophyte treatments over time. And we see there's a relationship between diversity and ecosystem stability. So the figure you see here is Shannon's diversity, which is based on the taxonomic data set. Along the x-axis is time, and on the y-axis is Shannon's diversity index. And we see that taxonomic diversity was highest in the high macrophyte treatment. There was a drop in diversity. The drop in diversity in the no macrophyte treatment corresponds to that peak in biovolume, which is namely chlorella. So I can also run diversity measures on the functional data sets. I had the resource acquisition and the herbivore interaction. In general, if we just look at all three plots, we see that the diversity matrices show a tendency for greater stability in the high macrophyte treatments, the green. If we look just at resource acquisition, functional diversity increased in all treatments, which says that that there's an increased diversity in the abilities of taxa to acquire nutrients. Alternatively, the overall herbivore interaction decreased from day 1 to day 76 in all treatments, give or take, and this suggests that communities are more similar in ways to avoid predation. So perhaps there's some sort of selection pressure by zooplankton that taxa can evade predation. I also ran the same principal response curve analyses for the functional traits to determine if there are certain functional traits that had a strong response, similar to how I did with the taxa. And this is one of the results. We see that prey quality had a strong positive response. This is graphing showing the proportion of excellent prey quality, which is based on the fatty acid content of a taxa. And it's showing that it's generally higher in the high macrophyte ponds. So we can see if we compare this to overall how the phytoplankton biomass changed over time, which is in the figure on the right, we see that although the phytoplankton biovolume is low, the prey quality is excellent for our zooplankton. However, another important trait that came out from the principal response curve analysis was the maximum length, or we can call this the greatest actual or linear dimension, GALD. And that red line in the figure on the left is at 35. And when the community is represented more by taxa above that red line, that is the inedible portion, and below is the edible. So although the prey quality was excellent, the phytoplankton communities were shifting to become mostly inedible to zooplankton. And this is bringing to the bigger picture. The middle panel is showing metaphyton, which is sort of like a filamentous green algae that is also inedible. And this increased over time in our experiments. And the final panel is the zooplankton biomass. And so if we put this all together, we start to see that our communities are shifting under this nutrient press to be populated by taxa that are inedible. And so if we go up the food level to the zooplankton, could this mean that our zooplankton population will decrease if there's not enough edible phytoplankton under this continuous nutrient press? So the phytoplankton are telling a really interesting story of what happened along the way of this experiment, and then we get to day 76. We've looked at the taxonomic and functional analysis to provide insight. And at this final day 76, we also have bacteria data to add to this information and improve our understanding of what's going on. Studying bacteria in addition to phytoplankton provides a clearer view of the functioning of shallow lakes. The more we know, the better our perspective and the better we can assess the system. Sort of like if the Human Genome Project only studied one microbe, then they would come to a different conclusion of the body's condition on the role of that one group. Or even more simply, if you've never looked under the hood of a car, you might think that all a car needs to work is a key. But after some days in the garage, you'd learn that there needs to be a key, an engine, a battery, and I can go on. Each component has a role to the overall functioning of the car, and without something, it could work, but it might not work efficiently, with quality, or for very long. So bacteria play important roles in shallow lakes in many biogeochemical processes, such as nutrient cycling and decomposition, which is not done by phytoplankton. Here in this figure, they're circled in yellow, and they interact with nutrients, macrophytes, phytoplankton, and zooplankton. Knowing about the diversity of bacteria in these ponds could speak to the ecosystem stability and abilities. So I have bacteria samples from one date, which I processed from the two habitats, from the free living and associated with macrophytes. I got information on the taxonomic communities by sequencing a highly conserved gene in all bacteria. And although it's highly conserved, there are differences in the gene which allow me to classify the bacteria taxonomically so I can study diversity. So this is a figure of the relative abundance of different bacteriophyla in the macrophyte treatments and the two habitats. The y-axis is the relative abundance, and there's a bar for each water sample. And I know that there's a lot of bright colors. And so there's no need here to try and distinguish between the orange from the orange-yellow, but instead to look at the bigger picture differences. A big difference can be seen when you just compare the attached to the free living communities. The attached community has a whole other phyla present, that blue color, distinct from the free living. They also have some pink, which is acidobacteria, and higher abundance of cyanobacteria, which is the green. So it appears that the differences between habitats, free living and attached, is greater than the difference among macrophyte treatments within a habitat. And then we can start to question, are these differences significant? Or are we just overwhelmed by all the colors? Well, these differences are significant. In this figure, I measured Shannon diversities in the two habitats. And we see that the Shannon diversity in the attached habitat is much higher, significantly higher than the diversity found in the free living habitats. This is all free living samples. And we can look at the difference between macrophyte treatments in one of the habitats. So this is showing the Shannon diversity between the three macrophyte treatments in the free living bacteria communities. And there is no significant difference. However, we see that it appears that the high macrophyte treatment has a tendency to have greater diversity in bacteria community. So it's possible that these communities could have similar measures of diversity or similar amounts of diversity, but actually have different community structures. And so to answer this question, or to figure this out, we can compare the individuals in communities. And that is our next figure. This is a Venn diagram of the taxonomic data for the free living bacteria in the three macrophyte treatments. For bacteria, we work with OTUs, operational taxonomic units, which are like taxa-like units. And I will most likely just call them taxa for ease. So if we were to divide all of the OTUs that we saw in our samples among the three treatments, only 21% would be shared by all three treatments. Whereas 18% are unique to the high macrophyte treatment, we have 11 which are unique to the low macrophyte treatment, and 17 which are unique to the no macrophyte treatment. We also see that similar to the phytoplankton data on day 76, the low and high treatments are more similar than the high and none and the low and none communities of free living bacteria. And so from this analysis, we can do perform a more accurate analysis, which takes into account the abundance of these OTUs in the ponds to determine, are there certain taxa units which are indicators for a certain treatment. And this is our indicator analysis. The indicator taxa are units. The indicator taxa units are only found in one macrophyte treatment, and they're present in most or all of the replicates of that treatment. And in this figure, we see that the high macrophyte ponds has the most amount of indicator taxa units. It has, or OTUs, sorry, it has 218. In the none, there's 132 indicator OTUs, and in the low, there's 37. And I have colored them to be representative of the phyla. And so in the high macrophyte treatment, we see a lot of the indicator OTUs are from the proteobacteria phylum, In the none, there's a lot of acidobacteria. And bacteria at this phyla level, there's still a lot of diversity. So in the no, there is a high amount of unique acidobacteria, but those acidobacteria are like the most abundant and widespread phylum. So it's hard to, at this level, to say more about those indicators. Similarly, there's the high amounts of proteobacteria and the high macrophyte treatments, while proteobacteria are a very metabolically diverse phylum, and so I can't speak to a certain function that those indicator taxa are doing. The takeaway here is that we do see that under nutrient addition, macrophyte abundance had an effect on the bacterial communities. The higher amount of unique indicator OTUs and the high macrophyte treatment can be mirroring that the same high overall phytoplankton diversity we saw at the taxonomic and functional level. And so, to put all these findings together to make the story of the community response in ponds, we see that there's a lot of essential interactions at this microscopic level. The macrophytes increase, ponds with higher macrophytes generally had higher diversities in both phytoplankton and bacteria. And we can infer that there was interactions between the phytoplankton and the nutrients that the macrophytes were inhibiting. So we have more, with this research, we have more insight into the dynamics driving microorganism communities. And as a result from these experimental ponds, and the results from these experimental ponds can be applied to responses in shallow temperate lakes, leading to increasing our understanding of mechanisms driving ecological processes that regulate and modulate ecosystem responses to external changes in these essential freshwater systems. There's always more to this. There's always more research to be done and more to this story, but we will save that for another time. For now, I just want to say thank you to my supervisors, my community members, and my many collaborators, and for everybody for listening. Thank you.